Learning Objective 3 is about sensory receptor fields. Let's first define receptive fields. So a receptive field is corresponding to an area of innervation where individual mechanoreceptor fibers convey information from a limited area of that skin. So essentially the field out of which a nerve fiber is receptive. If you think about it, that kind of makes sense. The name of it makes sense. They can vary in size from very large to very small, depending on the area of the body that you are located in. And if you think about it, that really makes the skin essentially a complex mosaic of spots dedicated to specific sensations. Now, these areas of innervation from individual mechanoreceptor fibers are usually so small that we don't particularly notice that, hey, if you poke right here, I feel pain, but right next to it, I only feel vibration. It's just so small, it's beyond our kind of conscious awareness of it. But at a micro level, that's essentially what it is. One way that we test the function of receptive fields and innervation fibers is through the two-point discrimination test. It allows us to use it as a diagnostic tool of peripheral sensory deficiencies. Where the receptive fields are the smallest, tactile acuity is the highest. This means we have the ability to detect very small differences in sensation. And then conversely, in the largest receptive fields, tactile acuity is the lowest. So what that tactile acuity means is that, again, we have the ability or not to detect or sense consciously very small or large differences in sensations. Now, the way that this test works is by using a tool, a two-point discrimination test tool, such as this, or you can even use a paperclip if you bend it in exactly the right way. And what happens is there are these prongs. The prongs are spaced a certain number of millimeters apart, which is shown on these little discs here. And you ask a patient or the test subject to close their eyes, and then you touch the two prongs spaced a given measurement apart, millimeters apart, at the same time on the surface of the skin, such as what is shown in this right image here. And you ask the person, do you feel one prong or two prongs, or 1 point or 2 points? And if you are touching a single receptive field, so say hypothetically, these two prongs on this finger is touching one receptive field, then the person would only feel one prong. And if they are touching two different receptive fields, then they would feel two prongs. Okay, that kind of makes sense. So what this means is when we say tactile acuity is highest in the fingertips and lips, for instance, we're looking at a fingertip here, it's highest because the receptive fields are so small. And in this case, if let's say this test is using a distance of, let's say 4 millimeters between these two prongs, it's touching two different receptive fields. And so you consciously detect two prongs. Now, there is a threshold, particularly on the fingertips. If you get down, I think the threshold is about 3 millimeters or so, but really 2 millimeters or even 1 millimeter, you won't be able to feel two prongs anymore. So go ahead and test this on yourself. It's kind of a fun exercise, or test it on a roommate or something like that. And ask them to close their eyes and tell you, do you feel one prong or two prongs? And you'll notice that when you go to areas where the receptive fields are larger, say on the back of the arm, for instance, the distance at which that they are able to detect the two prongs will grow. That means that the prongs will have to be spaced farther and farther apart as you go into areas of lower and lower tactile acuity. So here's a fun experiment for you to try out at home. It is kind of fun. It's interesting to see what your tactile acuity is and how far apart you can feel those prongs as two distinct points on the fingertips, back of your arm, and then say the thigh or the top of your shoulders or your back where the tactile acuity is the lowest. And then think about this. Why do we need such fine discrimination of sensation in areas like the fingertips or the lips? Well, fingertips make sense because we do very complex fine motor movements. We need that ability to sense what it is we're touching with our fingertips. And I would argue also with the lips, particularly when it comes to things like eating. We need to be able to detect this via our sensory nervous system, very fine details in what it is that we're eating. So that is why tactile acuity is highest in the fingertips and the lips. And importantly, this test can be used to get a sense of the scope of peripheral sensory deficiencies and where the sensory deficiencies occur. Learning objective 4 is about the brain processing of the somatosensory system and its input. It is very important to recognize that physiologically, we physiologists like to differentiate and categorize areas of the brain by their function, rather than purely by their anatomical delineations. This can make for some fun conversations, and hopefully this isn't too confusing for you. But in general, please let me take an opportunity to go over that when it comes to physiological cortices, we like to describe them in terms of being the primary cortex, the secondary cortex, or an association cortex. And you'll find that there will be an ongoing theme, and we'll talk more about these when we do special senses in the final section of the neuroendocrine course. So what I'm going to talk about next is the primary, secondary, and association areas that are involved in processing the body senses. This is the primary somatosensory cortex, abbreviated S1, or the primary could be a 1 with a little degree symbol after it. Anatomically, the primary cortex is located in the postcentral gyrus, and it is the first stop for most cutaneous senses, meaning that when it comes to input coming up from either the lower brain stem or the spinal cord, this is the first stop for processing of those senses. And what happens as a result is that these senses that are processed here are very crude. Okay, so this is more about determining the position of something, its size, or even some ability to discriminate shape, such as a sphere or a cube or a pyramid shape, basic shapes. When it comes to the homunculus, there remains somatotopic representation, which is what's depicted by the famous homunculus image that is overlaid across the primary sensory cortex, meaning that this representation is proportional to the input from the body and what peripheral anatomical structure that it is innervating remains, and it kind of maintains that proportionality. This is the secondary sensory cortex, often abbreviated as a 2 with a degree symbol. Now, the secondary sensory cortex is located in the wall of the sylvian fissure, also known as the lateral sulcus, and it receives input from S1, meaning, remember how I said in the previous slide that the arising somatosensory information will have its first stop at neurons on the primary somatosensory cortex. Well, these neurons will send their projection on to the secondary sensory cortex, where they will have synapses there for more processing. Now, primary sensory cortices and primary cortices in general are more involved with crude functions, but secondary cortices are often more elegant. They are more sophisticated in their processing. And that's true for the secondary cortex as well. We say that the secondary somatosensory cortex is responsible for the interpretation of what we call cognitive touch, as opposed to for the primary, which is crude touch. Okay, secondary is about cognitive touch, meaning that you're able to really make some executive higher level comparisons and interpret what you're feeling. And that's exactly what this box down here is representing. Comparisons between objects, maybe determining which object in your hand is heavier or smoother or has a greater texture or bigger. Different tactile sensations and identifying them according to textures, determining whether something becomes a memory as well. So as you can see, this is more about cognition or higher level thought processing about what we are touching, rather than the mere input of touch itself. Now this is the last slide that really I'm going to talk about when it comes to the brain regions involved in the interpretation of senses. And this is what I mentioned a few moments ago about the very high level of a bunch of different types of association cortices coming together to do very complex thinking. So this is not a brain region that you really learn about anatomically, because it's really three different areas. It's the parietal brain region, the temporal brain region, and then the occipital brain region. But at the junction between those three cortices, the parietal, temporal, and occipital cortices, we have a very important physiologic brain association cortex called the PTO, or the parieto-temporal occipital cortex. It is, as I mentioned a few moments ago, an association cortex. So it doesn't have that kind of big job that the primary sensory cortex does. And it doesn't really have the somatotopic organization of a secondary sensory cortex. And in fact, it doesn't really have that much somatotopic representation at all. It's kind of a jumble. But what it does is it does very advanced thinking, if you will. Now, more formally, we call this a high-level interpretation of sensory inputs. So you get sensory input. It goes to the big job area first, the primary sensory cortex. Some information gets sent to the secondary sensory cortex for some processing. And then from there, the information gets routed over to the PTO, not only from the secondary sensory cortex, but from a lot of different primary and sensory cortices. For instance, the visual cortices, integrating with movement and the ability to see and feel, proprioception. All of these things really come together at the PTO. Not only does it, or as a result of receiving input from the multiple sensory areas, as a result, it's able to do complex analysis, not only of what it is you're touching, but yourself in relation to your environment. For instance, the PTO is involved in the identification of objects, as well as analyzing coordinates in your environment. Something's in front of you, above you, below you, etc. And it's just really cool because it is able to do all of these functions, and it does so because it gets sensory input from all these other association areas. Okay, so if we were to break this back down, we could say I pick up something in my hand and it's heavy and it's got a smooth surface. Okay, that's kind of a primary sensory cortex type of thing, big job thing. Now, secondary sensory cortex could say, okay, it's heavier than the thing in my other hand. So I know that they feel the same, but this one's heavier, so that must have something inside of it. Now, that information, if I were to use my eyesight and my memory and remembering things and naming things, I can look down at the thing in my hand and realize it's a cup of water. And my other hand is holding an empty cup of water. So that's kind of the difference between major crude information, a little bit more advanced information, and then this very advanced interpretation of sensory inputs.